welcome to Virtual Darwin Days. Thank you so much for joining us for our programs today. My name is Laura Beth. I'm going to be your host for this program, and I'm so excited to talk about cheetahs, one of my favorite animals, and I'm sure y'all are excited too. We do have folks joining us on YouTube and on Zoom, and before we get started, I'm going to do a quick Zoom tutorial just in case this is, your first time to this platform. So your screen might look something like this once we share our screen and share the PowerPoint presentation. We do ask that you stay muted during the program and leave your video off. That's just for your privacy. But we would love to hear from you, whether you're in YouTube or on Zoom. Please use the chat to ask your questions and leave your comments. And we do have captions available on YouTube and in Zoom. In Zoom, to enable them, you just click that button that says CC at the bottom of your screen, and then go to show subtitle. And you can change the way those look by going to subtitle settings if you'd like to. And you'll also want to make sure that you're in speaker view, and you can choose what's called side-by-side mode. So that will show the PowerPoint presentation beside the video. And then you can use this nifty little line here to make the video bigger or make the slides bigger, whatever you choose. So I'm going to go ahead and introduce our expert for today, Morgan Maly. And Morgan is a PhD candidate in genetics at North Carolina State University. She's also a graduate researcher at the Smithsonian Conservation Biology Institute's Center for Species Survival and Center for Conservation Genomics. And I wanted to give you a little more background information about Morgan just because her experiences are just super cool, as you will hear. So Morgan has a long history of working with animal poop, from her early days cleaning up after dogs and cats at veterinary hospitals, working with dogs at the Iditarod in Alaska. She's also worked at wildlife rehabilitation centers, and of course, she has lots of research experience, including studying fecal samples from giant anteaters in zoos. She helped set up a non-invasive fecal hormone monitoring lab in China for the giant panda. And she's worked with scat dogs, tracking down cheetah fecals in Kenya. And so her current poop project is focused on understanding dietary factors associated with gastrointestinal disease in managed cheetahs. So with that, I'm going to go ahead and turn it over to you, Morgan, and thank you so much for being here. Super excited to learn more from you. So take it away.
Thank you so much for that introduction. Yeah, I'm very excited to be here today. Let me share my screen with you and we will go ahead and get started. So as you guys have questions, please feel free to throw them in the chat and I'm happy to talk about them as we're on the slides. We don't have to wait till the end for that. As Laura Beth said, my name is Morgan and I'm here today from NC State and from the Smithsonian Conservation and Biology Institute, which is a bit of a mouthful, so you might hear me shorten that to SCBI. And I'm going to talk to you a little bit about my PhD project today, which is looking at poop of cheetahs to help better understand their dietary needs and health. So we'll go ahead and get started. And before we get into the poop science, I wanted to go over some fast facts about the cheetah. And so many of you probably know that the cheetah is the fastest land animal, and it can clock speeds up to 70 miles per hour. However, I think it's really important to see just how fast a cheetah is. So I wanted to show you a simulated video. It's got kind of some funny graphics, excuse me, but it really puts in perspective just how fast a cheetah is. So we're going to watch a 100-meter race. And you're going to see a simulation of the world's fastest cheetah. Her name is Sarah, and she ran this distance in less than 6 seconds. She's also going to be racing the world's fastest human today, and so that's Usain Bolt, and he ran the same distance in just over 9.5 seconds. And then just to really put it in perspective, they're also going to be racing against an average human. And so usually it takes normal people about 15 seconds to complete this distance. So I just want to show you really quick the simulated video, and you can see right away the cheetah is off to an early lead. So cheetahs can go from 0 to 60 in just under 3 seconds. And so it really leaves all humans in the dust. It's already done with its race. World's fastest human comes in, and then eventually here comes our average human. And so I think this really does a good job of putting all three of these speeds next to each other so you can really get a new respect for just how fast cheetahs are. Okay, so we've just seen the incredible speed that the cheetah has, but what makes it able to achieve these speeds? So cheetahs have many adaptations that make them built for running. And so in this slow-mo video that I took, this is from the Cheetah Conservation Fund in Namibia. You can see that cheetahs have these really long, slim bodies and these very flexible spines. And so this makes them very aerodynamic. And so they can easily run fast. And you see they have these long, lean bodies with thin legs, smaller heads, and really big back leg muscles. And if we compare them to other big cats, so if we look at a lion or a leopard, you see that these cats are much more muscular and thicker bodied with bigger heads. And so these big cats are actually built for leaping and jumping, whereas the cheetah is built for running. Some other features that cheetahs have include non-retractable or semi-retractable claws. So if you think about these big cats, or maybe you have a cat at home, they can often put their claws out and then they can bring them back inside of their feet. Cheetahs do not do this. So their claws are out all the time. And this helps them in running in that they can use them much like cleats to help keep traction on the ground. Cheetahs also have these really, really long tails that they can use as a type of rudder. So as cheetahs are running at these really high speeds and they need to turn really quick while they're chasing their prey, they can use their tail as a type of counterbalance to keep them from falling over. Cheetahs also have really large nasal cavities, and this helps them get increased airflow to improve the amount of oxygen that gets into their body so they can run at these high speeds. And so you might have noticed that a lot of these characteristics are really unique to cheetahs, and they don't apply to most other cat species. And so another unique aspect to cheetahs is that they are daytime hunters. So most other cats choose to hunt at night. So some adaptations that cheetahs have for hunting during the day include much larger eyes. So cheetahs actually rely more on their vision and less on their smell than other cats. And so this is why you often have that picturesque scene of a cheetah sitting on top of a termite mound. It's often surveying the savanna, looking for its prey. And so they rely a lot on their vision during the day. They also have these quintessential black tear marks here. And so these are believed to help reduce sun glare, much like you might see athletes out on the field at like a football game, putting black marks under their eyes to reduce the sun glare. And so cheetahs have developed a lot of these adaptations to help them run really fast. Other than knowing that cheetahs are fast, you might have also heard that cheetahs have very low genetic diversity. So what does this mean? All living things are made up of DNA, and we all have a genetic code. And those are made up of those 4 letters, A, T's, C's, and G's. And so the genetic code of cheetahs is actually really, really similar between cheetahs. And it's actually so similar that if we took a skin graft from one type of cheetah and donated it to another cheetah, that recipient cheetah would accept that skin graft even if the cheetah that donated it was completely unrelated, because the cheetah that receives that skin doesn't recognize that skin graft as a different individual. Their genetics are so similar that it thinks it came from itself. And so how did cheetahs get so low in genetic diversity? This can occur when a population becomes really small from overhunting, habitat loss, or climate shifts. And this can cause the population to crash and leave only a few survivors. And so we call this type of situation a population bottleneck. And so I wanted to illustrate to you today what a population bottleneck might look like. So we can imagine we have a large, healthy population of cheetahs. And so here the different colors indicate variation in that genetic code, or what we would call genetic diversity. And so if a catastrophic event occurs, something like a major climate shift, many of these animals will die off, and it would leave only a few animals that survive. And so these survivors are all the genetic diversity that's left for future populations. So we can see we already lost a lot of those different colors. So these survivors then give rise to the next generation, and then they give rise to the generation after that, and so on. And so again, all the genetic diversity that's indicated by these colors in this new population is limited by what these original survivors had. And so when you lose large numbers of your population, you have a really huge reduction in the genetic diversity. And this can be very bad for future generations. And so this is what happened to the cheetah around the time of the last ice age. And so it's also what's happening to many species now as they face extinction from humans and climate change. And so this is also why it's really important to maintain as much habitat for wild animals as we can, so they can keep their population numbers high, strong, and healthy. And unfortunately, many wild plants and animals are losing their habitat, and the cheetah is no exception. So we can look at this map, and if we look at these brown areas on the map, this is where cheetahs used to be found. So this is where they used to live before the 1900s. And you can see they covered much of the continent of Africa, up into the Arabian Peninsula, and even all the way into India. Now, if we look at those orange regions on the map, this is where cheetahs are currently found today. And you can see they've lost quite a bit of their habitat. In fact, they only live on less than 10% of the land that they used to live on. And so reasons for this include obviously habitat loss to human use. So this can be humans making buildings, farming, collecting resources like wood, or building roads. There's also conflict with humans because cheetahs are predators, so they're often seen as threats to ranchers and other humans. And then unfortunately for the cheetah, the illegal pet trade is a huge problem. So cheetahs are often, cubs are taken from the wild and smuggled for the pet trade. And unfortunately, only 1 in 6 of these cubs survive the journey. And even if they do make it to their destination, their lifespan is greatly reduced due to the lack of proper care as pets, because cheetahs don't make good pets. And so you can see there's a lot of reasons why cheetahs are in need of our help to ensure their survival. And so this is where different conservation initiatives come in. And so when most people think about wildlife conservation, the first thing that pops into their mind is a wild animal out on the savannah with some scientists sitting in a safari jeep, watching it and studying it with their binoculars or writing things down on a clipboard. And you're not wrong, this is definitely part of conservation. So much of conservation involves studying animals and their natural habitat. And this is where we can see where they go, what they're eating, how they're behaving with each other and other animals, and get the kind of information we need to help protect the most suitable type of habitats for these animals and prevent conflicts with humans. However, there's also another side to conservation, and this includes animals who live in managed facilities, such as accredited zoos like the Smithsonian or the North Carolina Zoo, or in museums like the one hosting these talks today. So the North Carolina Museum of Natural Sciences doesn't have any cheetahs, but they do house other endangered species that contribute to their conservation. And so animals in these types of facilities are what we refer to as insurance against extinction. Populations. And so if something drastic were to happen to wild populations, where their numbers became really, really low and they were too low to be sustainable, then we have these managed individuals in our facilities that can contribute their genetics to the wild populations. And so we can then increase the population numbers and the genetic diversity. And so animals in managed facilities are also much easier to study. You can imagine it's pretty hard to find a cheetah in the wild, let alone find that same cheetah multiple times. And so by studying cheetahs in a zoo, I know exactly where that cheetah was born, who its parents are, who its family is, what it's eating. And so there's a lot more variables we can control to really get down to the basic science. And so the best approach to wildlife conservation is really to do both types of these conservation. And so each population has its own conservation challenges, and often studying one of these populations answers questions that helps the other population. So as I mentioned, each population of cheetahs have their own challenges. And one of these challenges include gut health. And specifically today, we're talking about a disease called gastritis. And so gastritis is essentially inflammation of the stomach. And in cheetahs, some symptoms that we see are vomiting, diarrhea, loss of appetite, weight loss. You can see none of these symptoms are very pleasant for the individual, and they actually can be quite detrimental. And so when we look at our managed populations, unfortunately, we do see that this disease is really common among our managed cheetahs, and it's actually one of the leading causes of death. However, if we compare this to cheetahs in the wild, we rarely find cheetahs with gastritis. And so this makes us wonder why this might be. And so as scientists, when we see situations like the difference in a disease between these two populations, we think about what might be driving these disease differences. And so our first step is to identify all the possible differences that exist between these populations and test whether they might be influencing this difference in disease. And so this is essentially the basic formula for building a hypothesis. And so that's exactly what we do. And so some of the hypotheses that scientists have come up with are there might be some differences in bacterial infections between the managed and the wild cheetah populations. There might be differences in stress or chronic stress. So living in close proximity to humans or under human care might cause a specific kind of stress that makes cheetahs more vulnerable to gastritis. And there's also this difference in diet. And so cheetahs in the wild are eating very different meat than what cheetahs in captivity or managed facilities are eating. And so there's been a little bit of research on the first of these two hypotheses, and I decided to focus more on these dietary differences. So if we think about diet differences between wild and captive cheetahs, we know that cheetahs are cats and cats are carnivores. So they both are eating meat, but it's not necessarily the same kind of meat. So what do I mean then by the dietary differences? So on the left here, this is a pretty typical zoo diet. And you can see these cheetahs are eating a ground meat, and this is commercially available. This is pretty similar if you have cats at home in a can and you feed them canned food. It's like a nice pate. And so this food has been formulated with the right amount of protein and minerals and vitamins, but it's a very lean meat. So that means it's really easily digestible for these cats. Now, if we compare that to what cheetahs in the wild might be eating, cheetahs in the wild are obviously hunting for their own food. And so they're eating from carcasses, right? And so when you eat from a carcass, you have to tear through things like fur and skin or feathers. And you're also eating things like ligaments and bones and tendons. And so it turns out that these harder to digest parts of the carcass are acting as a type of fiber in the cheetah. And so much like you and I need fiber in our diet, so too do cheetahs, but it seems that they might be adapted to getting it from a different source since they are carnivores. So why do we care about fiber? So you've probably heard from doctors, your parents, or parents, maybe you've even heard it from your kids, that fiber is important in your diet. But why? So let's think about first where fiber comes from. So you and I, we're humans, we're omnivores. That means we can eat plant material and meat, right? And so our fiber comes from plant material. So this is usually green leafy vegetables, fruits, and whole grains. Now, cheetahs we discussed are carnivores, and so they can't digest plant materials. Their gut is not built the same way. And so as we discussed, we think that they're getting their fiber from those harder to digest parts of the animal, right? And so now we see where this fiber is coming from, but again, what is it doing? Why do we care how much fiber we're eating? Well, fiber gives us a lot of benefits, but I'm going to simplify it down to just a few things. So the first is that fiber increases production of good fatty acids in our gut. And these fatty acids are essential for different processes that we do in our body. And so our bodies are able to produce some of these good fatty acids, but actually microbes are much, much better at it. And so they can ferment these harder to digest parts of the plant and make even more good fatty acids. And so a lot of these good fatty acids in our gut are coming from microbes fermenting that fiber and turning it into fatty acids. So by eating more fiber, we feed or cultivate a larger diversity of these good microbes, and that can increase the production of those good fatty acids in our gut. But what exactly are these good fatty acids and microbes contributing to? So having a balanced set of microbes in your gut is really important. So from many studies in humans and in mice, we know that microbes in our gut are heavily involved in digestive health. They help us process our food and protect our gut from bad microbe invaders. And so this makes sense because after all, these microbes are living in our gut. So it makes sense that they would affect our digestive system. However, we've also learned that the gut microbes are really important for the immune system. They help with things like training the immune system on what a pathogen or a bad bacteria is. And there have been many associations with autoimmune disorders and abnormal gut microbe communities. There's also, oh no.
Oh,
it's okay. There we go. We're fine. Okay,
hey,
sorry.
Morgan, do you mind if I ask a quick question?
Of course.
I hope it's quick. Okay. Can cheetahs get autoimmune disorders that we know of?
So I don't know off the top of my head of any specific ones, but we do have a lot of like chronic inflammatory issues, which are usually classified under some type of autoimmune disease. So we think that's probably where they fall in that realm.
Gotcha. Okay. Thank you.
Yeah, no problem. Good question. Okay. So the last thing on here is the central nervous system. So we do also see associations between gut microbes and the central nervous system. And actually, we've seen a lot of associations with behavioral and brain-related disorders like Alzheimer's, autism, and even depression. And so we can see why the gut microbes are really important to study. And we often study them as a community because there's more than one type of microbe in your gut. And scientists like to call this a microbiome. So you've probably heard the term microbiome before, maybe in the news. So I just want to make sure everyone knows exactly what we're talking about. And I think it's actually easier to first think of like a larger system. So a lot of people have heard of maybe an ecosystem or a biome. And many of us are familiar with this concept of different plants and animals and invertebrates all living together in some type of habitat, whether it be a forest or a savanna. And then, of course, we have to think about all the different interactions between these individuals, right? So that's what a biome is. That's a larger scale. Now, a microbiome is essentially the same idea, but micro, so shrink it down, right? And now, instead of a habitat like a forest or a savanna, that habitat is now you or your gut. And instead of plants and animals and invertebrates living inside you, you have things like bacteria, viruses, and fungi. And so these guys are the main players in the microbiome, and they also all have their own set of genes. And so these different genes, part of their genetic code, allow them to perform specific functions in your gut. And then, of course, again, we have to consider all the different interactions between these guys and their habitat, which is you and your gut. And so it might be weird to think about microbes living in your gut, but don't forget that your entire body is covered in microbes. So before you get alarmed, just remember that most of these microbes, almost 99% of them, are neutral or beneficial to you and not harmful. And in fact, most of these microbes help fight off bad microbes or pathogens from taking up residence on your body. But if something is off in your microbiome, then you may no longer be protected from those bad bacteria. And so this is why scientists are really interested in studying microbiomes, so we can learn more about how these different communities work together and how they protect their hosts, AKA you and me. And so there's roughly 5 major regions that are studied in humans, and these include the gut microbiome, which we've just talked about. They also include the urogenital region, your airways, so your lungs and your nasal passages, your skin, and your mouth or your oral cavity. And so we're beginning to learn a lot about these microbiomes in humans, but there's still a lot we don't know in other species, including the cheetah. And so the project I'm presenting today is starting to fill in some of these gaps, particularly about the gut microbiome. Okay, so for my project, we developed a hypothesis about how the diet influences the gut microbes and then how those gut microbes affect gut health. Because remember, we said they had different kinds of diet. So if we imagine a cheetah in the wild, it's eating that very high fiber diet. So we can assume that then those cheetahs would have a much higher diversity of microbes in their gut. And then that would mean that their gut health is far more improved. If we compare that to a cheetah eating a really low fiber diet, we then might assume that they have a much lower gut microbe diversity. And in turn, this means that their gut is not as healthy. And so fiber may affect the microbes in our gut, but what does that actually look like? So to measure the gut microbes, we often collect fecal samples because they're much easier to collect than trying to sample the inside of a cheetah gut. And poop is really all the stuff that comes out of your gut, and that includes the microbes that live there as well. So here we have our 2 different diet types and our fecals, and now we're going to talk about what we might find in those fecals. So here we have a fecal sample, and all these different shapes and colors inside the fecal represent different kinds of microbes. And so you can see there's not a whole lot of diversity here, right? There's only a few different kinds. And so now let's compare that to a poop from a cheetah eating a high fiber diet in the wild. So here we can see this poop has many more different shapes and colors, indicating that there's a greater diversity of microbes. Now, these are 2 very extremes, very low and very high, but in reality, zoos actually feed kind of on a spectrum. And so even zoos that do feed this low fiber diet often supplement with other sources of fiber like whole prey, like rabbits or bones. And so we really have to think about dietary fiber on a range. And so the Smithsonian feeds this low fiber diet, again, because it's nutritionally balanced, but we also add in whole prey items like whole rabbits, and they get long bones from things like cows and horses. And so we can imagine if there's a range of fiber in the diet, we can also hypothesize that the microbes found in their poop samples would also fall into a range between these 2 extremes. And so if we were to look at the microbes in these fecals, we can see that as we start to add fiber to the diet, the microbes start to look more and more like this wild cheetah poop. And so part of our research is to determine how much fiber cheetahs need to keep their gut microbiome healthy and happy. And so I'm also interested in identifying potentially pathogenic or bad bacteria that might be found in the gut. And so I'm indicating this here by this little yellow spiky guy. And so in animals with a healthy, diverse gut microbiome, these bad bacteria might not stand a chance at surviving and multiplying in that cheetah's gut. But in cheetahs that have low microbial diversity and not a lot of other microbes to compete against, these bad bacteria might have an advantage and grow to really large numbers in the gut that could cause illness or disease in their host. And so these are the questions that I'm looking to answer with our research. And so to start testing that hypothesis, many scientists first do something called a pilot study, and that's really just a small version of a big research study. And so we do that just to kind of see if it's worth moving forward. And so we did a small pilot study just to test the waters a bit. And we used 2 locations with cheetahs eating 2 different diets. And so the first location was at SCBI, and we are in Northern Virginia. And as I said, our cheetahs eat this low fiber, lean meat diet, and then they're also supplemented with bones and whole prey items like rabbits. So they're still getting some of that fiber material. We compared these to some cheetahs in Namibia at the Cheetah Conservation Fund. And so Namibia is at the southern tip of the continent of Africa. And these cheetahs at CCF are wild born, but they are brought into CCF usually to be rehabbed and rehabilitated and re-released. But some of these individuals are not able to be released. And so then they spend the rest of their lives living out a very luxurious life at CCF, excuse me. And so the cheetahs at CCF, their diet is entirely carcass meat, and most of it is donkey and horse, but occasionally they do get some wild game. And so we would consider this a higher fiber diet than those at the Smithsonian. So now that we have our locations and identified our cheetahs, we need to get some poop samples. But how exactly do we collect poop from a cheetah? So for the study, I wanted my fecals to be really, really fresh, because if a fecal is sitting in the field too long, all the microbes from that field are going to crawl all over the fecal sample, and then it's not going to look like a cheetah gut anymore. It's going to look more like the dirt from the environment. And so it turns out the best way to get fresh samples is just to sit and wait for them to happen. So usually when I tell people what I do, cheetah conservation, they think it's really glamorous and I'm working with cheetahs and get to travel and do field work. But the reality is I do things like this, where I just sit in a chair in 90-degree heat and just wait for a cheetah to poop. And so it was very, very hot this day. You can see there's a litter of cheetahs I was watching, and they are just very comfortable in the shade, staring back at me, wondering what the heck I'm doing sitting out in the hot sun. But eventually one of them does decide to get up and use the restroom. And so this is when the real excitement starts. And so working with the keepers, we make sure I can safely get inside that cheetah enclosure so that I can go collect my poop sample. And sometimes it, sorry.
Well, I was just wondering, do cheetahs have like a poop spot in their enclosure?
Yeah, sometimes they do. They usually have like a favorite spot. A lot of times they'll go up on like a high point. Like you can see this one likes to poop on top of this box. There are some mounds that we have that we call poop mounds. Some have a tree that they like to poop on. So they do usually try to go in the same spot, but sometimes they make it a little harder for us.
Okay.
Good question though. Yeah. So sometimes they do put it in really not so easy to reach locations. So here, one of them dropped it in one of their weather huts. So I had to crawl inside of that. I also had one to climb up a tree and poop from the tree. So that was a very interesting experience.
Wow. That's cool.
Okay. So now we've collected our cheetah poops. So what do we do next? So we need to take these poops to the lab, right? Because we're interested in the microbes that are inside of them. So we have our fecal filled with microbes. And so the first step is to pull out all the microbial DNA from the poop. So I'm particularly interested in specifically the bacteria that are in the poop. So I'm going to pull out all the DNA from all the bacteria that are in that poop. Then I can take that DNA and I can get to sequence it, right? So bacteria have DNA, which also have genetic code. And so I can get that genetic code from our bacteria. Then we can use a bunch of really cool software programs. And basically we take those sequences and they tell us about the bacteria that those sequences belong to. So we can take these sequences and we can identify the type of bacteria that are in the poop. We can also use the sequences to estimate how many of each bacteria are in the poop. And we can even predict what functions these microbes or bacteria are performing inside of the cheetah gut. And so that's pretty much what we did with the microbes that we found in the poops of the cheetahs from these two different locations, eating these two different diets. So we want to compare what they look like. And so I'm just going to show you a little glimpse of the data comparing the gut microbes, specifically, again, just the bacteria of the two locations and diets that we looked at. And so this here is a Venn diagram. And so we have a circle for Namibia in blue and a circle for Virginia in yellow. And the first thing I want to point out is where the circles are overlapping. So this is what they share. And there are 85 bacterial groups that are found in both Namibia and Virginia cheetahs. And so this is good that they share some. It tells us we did something right, because since they're all cheetahs, we would hope that they would share some of the gut bacteria for that reason alone. So many humans have bacteria in common simply because we're humans. And so the same thing applies to cheetahs. And the second thing is that you probably noticed that the Namibia circle is much larger. This is because there are 121 bacterial groups that are found in Namibia that were not found in Virginia. And so there's far fewer bacterial groups that were unique to Virginia, only 36, right? So these 36 only found in Virginia, while Namibia had 121 unique species. This indicates that Namibia has a much higher bacterial count and likely means that they have higher species diversity of bacteria, which again, we would expect because they're eating a higher fiber diet. And so if we look at another measure of species diversity, which we call species richness, that's what we are measuring here on the y-axis. And again, we're comparing these two locations. So we can look at this and see that Namibia, again, has a much higher count of species richness, indicating that their microbes have a higher diversity. And so, again, we're remembering that the Namibian cheetahs were consuming more fiber than the Virginia cheetahs. And so that might be part of why they have higher bacterial diversity in their gut. So we also did some predictions of what the functions of these bacteria were doing inside of the cheetah gut. And this work was done by Megan Maxwell. She's a former undergraduate who was working with our lab at NC State. And she's also helped out on some projects here at the museum. So Megan found that some of these predicted functions of the bacteria were involved in different types of metabolism. And that makes sense, again, because these bacteria are living in the gut. So it would make sense that they would partake in our metabolism. And so I'm going to show you two graphs. And again, we're going to compare the two locations. So the yellow is Virginia and the blue is Namibia. And here what we're measuring on the y-axis, it says percent of sequences. And so what that means is that it's that the percent of the bacteria that we found in those samples and what percent of them are associated with this specific function. So here, over 0.4% of the bacteria are associated with this linoleic acid metabolism. And so Namibia has a higher percent of bacteria associated with this metabolism, while with this second type of metabolism, actually Virginia has more bacteria associated with that metabolism. So we don't know exactly what this means yet. And again, these are predictive functions, but overall, they kind of suggest that there are metabolic differences between these two populations. And this might be hinting at those differences in dietary fiber that we see.
And Morgan, I'm not sure you'll probably get to this, but we do have a good question in the chat from Katie.
Yeah.
So do you know if the type of prey would make an impact, or does it appear to be a whole carcass diet that is making a difference?
Yeah, so that's a great question. So likely, different types of prey are going to come with their own types of bacteria that are on the prey, especially given that these are in two very different locations. And so that's definitely a factor. But right now for the pilot study, mostly what we're trying to look for is just if we do see differences so that we can then piece out more if it's the prey, if it's the type of fiber, or if it's the location. But absolutely, yes. Good question.
Thank you. Yeah, continue.
Cool. Okay. So I just have a few more slides and then I'm very open to questions if people want to talk more. So the last graphy slide I'm going to show you, Megan also found some differences between the two populations regarding possible disease-related functions. And so unlike the metabolism slide, where in one Namibia was higher and then the other Virginia was higher, what's interesting about this is that in both cases, the percent of sequences or percent of bacteria that's associated with these disease functions, they're both higher in Virginia. And so this might be an indication of differences in the overall health or even gut health of these different cheetahs, because we see there's higher incidences of parasitic infectious disease functions happening and amoebic dysentery functions happening in the Virginia cheetahs. And so I'm just going to quickly wrap up kind of everything we've talked about today. And so what we did is we looked at the gut bacteria that differs between the US cheetahs in Virginia and the Namibian cheetahs at the Cheetah Conservation Fund. And we saw that those cheetahs in Namibia had much higher diversity of gut bacteria compared to the US cheetahs. We also see that there was a difference in the predicted functions of the bacteria, and really interestingly, they related to metabolism and disease, which are something that we're really interested in studying. So this tells us we might be on the right path with where we're going studying dietary fiber. We also know, based on much like we see in humans and other animals, that diet is really important for that gut microbe diversity, and it's likely also really important in influencing the gut health in the cheetah. And so this kind of brings about this idea that microbiomes should be part of animal management and conservation. And this is actually becoming way more common now with many, many species, especially as we start to look forward to reintroduction of species from managed facilities into the wild. We want to make sure they're well equipped with the right kind of microbiomes. And so, I'm continuing this work. And as I said, we only did those 2 locations. And because we can't really piece out if it's like parts of the diet or the locational difference, we're doing a much bigger version of that pilot study. And so we're adding a lot more cheetahs. We're over 100 of them now, and we're looking at multiple locations. And so we're also looking across that whole range of dietary fiber that I showed you, from the low to the medium and the high. And then we're also going to collect health information. So all the cheetahs that come from zoos where we know their life history, the keepers are filling out surveys that tell us what kind of health problems these cheetahs have, so that maybe we can match those microbes with specific health issues. And so hopefully by the end of this study, we'll know lots more about the effects of dietary levels, different levels of dietary fiber on the gut microbiome, and then how these microbes contribute to the differences in gut health that we see. And so with that, I'd like to thank you all so very much for your attention, and to the museum for having me here today. And I would be more than happy to take any more questions that you might have.
Yes, I know I have a lot of questions. Folks, go ahead and put your questions in the chat, and we'll make sure that we get those to Morgan. We did have a question / observation. It looks like you were prepared for this presentation with your shirt. Did you?
Yes.
Okay, that's awesome. I actually didn't even notice that, but.
So this is part of my soapbox, which many of my friends know well. Whenever people find clothing in stores that they think is cheetah print, it's not actually cheetah print. Cheetah print is just spots. And so I actually even have a little slide about this, if anyone cares.
Yes.
But so cheetahs really just have spots, whereas what you see in stores is usually a leopard print. And so I won't make you guess which is which, but if we compare this one on the left here is the cheetah compared to the leopard, and I think it's really hard sometimes for people. So the cheetah has solid spots, whereas the leopard have what we call open spots or rosettes. And so their spot is kind of a broken up circle, right? And then they have this gap in the middle. And so that's how you can tell the difference between a cheetah and a leopard print.
No, I agree. I think that's really interesting, and we have several people who didn't know that. So yeah,
Cool.
I think that's important, and I know that this is true for reptiles, where their patterns will be completely different between every individual, and I've heard that's true for like tigers and leopards as well. Is that true for cheetahs?
Yeah, so we can often use patterns of the spots to help us figure out which cheetah is which.
Very cool.
So yeah, they have unique spot patterns too. Good question.
Yeah. So Nathan wants to know, do the Virginia cheetahs have a higher incidence of gastrointestinal diseases than the Namibian cheetahs? Great question.
Yeah, so even the cheetahs that are kind of wild born and brought into CCF, we do consider them now managed cheetahs, but they are eating that entirely carcass diet. And actually those cheetahs do have very, very low rates of GI disease compared to the ones in Virginia and the US. And even the South African managed cheetahs, and in Japan and in Europe, this is seen all over the world. So not just in the US cheetahs either.
And I was curious if you could elaborate a little bit more on how you determine the function of certain bacteria. I know it has to do with, the illnesses that you see.
Yeah, so the functions, for you mean for like these different pathways?
Yeah, like for individual types of bacteria, you said you were looking at the functions.
Yeah, so these are predictive functions. So because when we're analyzing the DNA from these microbes, we're really only using, it's called a marker gene. So all bacteria have what's called a 16S rRNA gene. And they all have it, which makes it great because we can sequence that gene, but their genes are all different based on species. So we can sequence that gene and it tells us what species they belong to. And so we can use that species and what gene content we think they have to sort of predict what they're doing in the gut. So if we wanted to know for sure what each individual species is doing in the gut, we would have to do something more like shotgun, where we're actually going to sequence more parts of that bacteria. So this is predictive and we can kind of guess at what they're doing, but it's not as informative. So if we really wanted to know what exactly each bacteria was doing, we would need to do a little bit more work to figure that out.
Gotcha. Okay, thank you. And kind of on that note, Chris had a question. What predictions would you make for the Asiatic population of cheetahs?
Yeah, so we don't know a whole lot about those. There is a population in Iran, but there's about 100 to 200 cheetahs, and they're really, really hard to study given their geographical and political climate. So we actually don't know a whole lot about them. We can predict that they are probably pretty genetically, their variation is probably pretty low because their population is really small. I would hope that they're eating, they're still wild cheetahs, so they're probably still consuming high fiber diets. So I would guess that they don't have the same GI disease that we see in managed facilities. But we don't know exactly how long that population will be sustainable due to how closed off they are.
Gotcha. And Julie had a question. Would it be possible to give cheetahs in zoos probiotics or fecal microbiome transplants to help with their GI issues?
A great question. So yes, that's kind of where the start of this whole project came from, because there is a lot of evidence for that. They do it in humans, right? The problem for us is that we didn't know what a healthy microbiome really looked like in a cheetah. So we kind of know what it looks like in the wild, and we have now some idea of what it looks like in managed facilities. But we're never going to have our managed cheetah microbiomes look exactly like the wild, just because they live in different places and then they're eating different kinds of foods. But if we can get a good picture of what a healthy managed cheetah microbiome looks like through this study, through sampling the hundreds of cheetahs we're looking at, then we can get a better idea of what kind of microbes we do want to transplant. So before we just start transplanting, we wanted to make sure we were putting the right kinds of microbes into these different cheetahs. But that's definitely something we're interested in looking into doing.
That's a great point. And Nathan had a follow-up question. Is this a case where simply higher microbial diversity results in better health, or is there a way to directly relate certain fiber-related microbes to improved cheetah gastrointestinal health?
Okay, I'm going to read that one just for myself.
Yeah, no, that's okay. I know it's
higher microbe results in better health. So the reason this came up is because there was a study where someone actually took cheetah fecals and like inoculated different types of animal substrates. So they put the different types of bacteria found in cheetah guts into like different kinds of rabbit parts and different types of fiber to see what was happening. And so we have like a direct connection between the bacteria in the cheetah gut and what they're doing with these different animal parts. So we do think there is a direct connection. We don't have an identification of exactly which bacteria it is that's doing these different things, because unfortunately that study did not identify the microbes that were in those fecals that they added to the different things. But we do know there is a direct connection between the microbes in the cheetah gut and the functions they're performing on the fiber.
okay. And Nathan clarified, is the question like, does diversity of the microbiome equal good health, or maybe it's like specific microbes that it's missing?
Right. And we think it kind of could be a little bit of both, right? Because We do see that animals in the wild, generally, this is a trend we see where they are more diverse. And even in humans and dogs and other species that are studied, generally microbial diversity in the gut is associated with better health. So people with like irritable bowel syndrome or IBD or Crohn's disease or other gut issues tend to have a lower diversity. So usually lower diversity is fairly synonymous with lower gut health. But that's kind of where the microbiome world is going, is trying to figure out what those microbes each are doing.
Awesome. And we've got a question from Meredith. You mentioned trying to determine what a normal cheetah microbiome looks like. I know in people this is extremely difficult to do because there's so much variation from person to person. True. I'm wondering whether this might be a more attainable goal for cheetahs since they have less genetic diversity overall. So does decreased cheetah genetic diversity lead to reduced variability in their gut microbiomes, making it easier to determine a normal?
Yeah, so that is one of our thoughts because cheetahs are so similar. We do know that microbes in the gut, right, interact with the genetics of the host. And because there's not as much variability there, we do think a lot of it is driven by the microbes. And also with humans, it's hard because we do eat such a variety of different types of foods, and we have different environmental exposures. And that's a little bit easier to control with the cheetah population as well, given that they're obligate carnivores and that they're all living in some type of managed facility. But yeah, that's a really great point.
Yeah, y'all are asking some really great questions.
I know, it's making me think.
Yeah. And my question from someone who does not do research at the museum, I'm an educator, but I'm just really curious, like, how do you go from a sample of poop to looking at the bacteria in that poop? Like, literally, what do you have to do? I know that's a gross question, but
Yeah, it's not the most fun I've ever had. So I'm just going to bring up that slide where I showed you this. So this step right here from going poop to bacteria or to DNA is a very, very long process. And usually what happens is this poop gets frozen. And so I have to cut up frozen poop because we usually take the center of the poop because it's the least exposed to the environment and to UV light. So sunshine can break down DNA and then it makes it hard for us to sequence it. So usually about 12 poops takes me almost 8 hours to cut up and take through the extraction process just to get the DNA out. And that's just one day for 12 poops. And then from there, I still have to do all the sequencing stuff.
wow. And I'm imagining like an apple corer, but
I really tried that with frozen fecal samples. The best thing I could find was using razor blades.
oh wow, okay.
Mm-hmm.
And so from there in the lab, you would, I assume, mix it with some kind of
Yeah, so
something.
there's kits they make and they go into different kinds of tubes and we use different reagents to kind of break. So bacterial cells are they're really well known for having some pretty hard to crack cell membranes. And so we use some reagents and enzymes that help burst open those bacteria cells so that they release their DNA. And that way we can then pull out the DNA sample from the bacteria. It's a very long process.
Interesting. Yeah. So yeah, how long is that process to, from actually getting like processing the fecal sample, how long does that take?
For one fecal sample, I would say it takes at least 2 full days to go from poop to a sample I can send to the sequencer. And I could do maybe 12 poops in those 2 days. And I have like over 100. So it's been taking me, and that doesn't include waiting for the poop and collecting the poop. And
Yeah. Which I don't know, I feel like waiting for the poop wouldn't be too bad. Like at least you're watching
yeah,
cheetahs have fun.
yeah, it's true. It's not the worst.
But so how did you get into this field?
Yeah, that's a great question. So I, as an undergrad, knew I always liked science and I liked animals. And so I think a lot of people in this like museum and research and zoo field can relate. And that, the only option we really thought that was there was to become a veterinarian. And so that's what I thought I wanted to do for a really long time. And I did a lot of internships and volunteering as an undergrad and after my undergrad. And so I did some that was veterinary based, and that's actually how I figured out I did not want to be a veterinarian. So I didn't like the aspect of, I love animals, and a lot of that is, taking care of very sick, or oftentimes, they don't make it. And so for me, I wanted to kind of step back and answer some bigger questions about why are these animals coming to the vet, especially when I was working with wildlife, and how can we kind of solve these problems on a larger scale. And so I originally was always into reproductive physiology. I thought like reproduction was really cool in animals, but I always liked wildlife. And so I happened to come across an internship at the Smithsonian that was looking at reproduction in cheetahs. And so I applied and I got it. And that's where I really started to fall in love with this idea of research and, different avenues to study the animals I like and help them, but without having to do it in a like one-on-one basis like a veterinarian does.
Mm-hmm. Wow. Oh, well, thank you so much for sharing all of your expertise with us.
Of course.
I imagine, on those days where you're in the lab using a razor blade on frozen cheetah poop, that you really have to think about the big picture, right? Like,
Yes.
this is ultimately doing a lot of good for these populations. So,
Right.
as someone who loves cheetahs, I will thank you for your service to cheetahs all over the world.
Thank you for making me remember why.
We do have time for one last question from Keter in the chat. Sorry if I mispronounced that. But what is the wildest thing that has happened to you thus far while studying cheetahs?
I'm thinking mostly about my field experiences. I don't have anything too crazy. There's a couple of close calls with animals in the wild, mostly with elephants that can be quite scary. And I also was stalked once by a wildebeest for at least a kilometer, but I was not worried about him because I was looking for fecals in the field, and so we're using the dogs to try to find them. And he would follow me and grunt, and then when I would turn around to like see who it was, I would see him, and he would like find the tiniest little tree or shrub to hide behind, but then still act all tough. So for me, it was like the field experience of just, being out in the wild and just knowing that anything out there suddenly could like be a little bit scary to you.
Yeah. Oh wow, that is crazy. I feel like I could hear more of those stories. But
Oh yes.
yeah, you don't have to share them all today. Well, thank you again for sharing your expertise. Everyone who joined, thank you so much for joining and sharing your questions. I really love learning more from you all and from Morgan. Before you go, I want to say thank you to our sponsors, our Friends of the Museum members who support us every day. And of course, we have an anonymous donor for Darwin Days who makes it all happen. So thank you so much. And we hope that you all can join us for the rest of our virtual programs today. We have an opossum talk coming up at 6 p.m. And then, of course, the Guess a Sketch, which is happening only on Zoom. So make sure that you register for that one. And if you're in the area, please visit us on Saturday at our downtown Raleigh location. We've got an outdoor event happening, lots of science activities and fun, more stuff about mammals. So we hope that you can join us then, and thank you so much for joining us now. We hope you have a great day. Bye. And bye, Morgan.